Yeah, thank you. Thank you for the invitation to present our work. Well, it's a continuation of the presentation of Rhiannon, or well, just some specific cases that we can show today. Novel strategies to assess drug safety combine advanced in vitro cellular systems with mathematical modeling. As Rhiannon has described, these microphysiological systems, organoids or other type of chips, well, they are usually human-derived, so they avoid cross-species differences in responses. They offer large experimental capacity. There is no tolerability problems with them. And well, we can really collect large data sets of high resolution and granularity. The problem they may have, one of the issues, is that the outputs in an MPS system is not straightforward to understand or to translate into patients' responses. And there is where mathematical modeling can help. We can help with the translation from an output that doesn't really it's not easy for us to see what it means in a clinical context to translate it into a clinical response. So the aim is to increase patient safety, and one of our main targets is to generate predictions prior to clinical trials, so that because there is where it's really important to mitigate toxicity before dosing the patients, and also reduce animal experimentation. So I'm going to speak about how we are assessing hematotoxicity and GI toxicity, because these toxicities are associated with cancer treatments, are very common, and what we are aiming at doing is developing dosing schedules so that we can mitigate the toxicities. We can find those schedules that maintain efficacy but minimize toxicity. So Rhiannon has explained this already. So I'm going to go very, very quickly. We have this bone marrow chip in which we have these two compartments. In one of them, we have a scaffold representing the bone structure, and it's there where we have mesenchymal cells to create the stem cell niche, hematopoietic stem cells, and all the bone marrow progenitors. And we sample from the other compartment, and there is a constant circulation fluid between the two compartments. So this already you have seen it in Rhiannon presentations, is how the chip, the scaffold has a similar microstructure to the human bone marrow. Also it has the same cell composition, and we sample the fluidics, the recirculating fluidics, and that gives us increases our sampling capacity, because we can keep a chip over time, so we don't need to use one chip for every sampling time. And this where it comes the translation. So what really we have in the chip are bone marrow progenitors, and we can measure how these progenitors respond to the drugs. And then what we have developed is a mathematical model that describes the human hematopoietic system. So it describes proliferation. It describes the bone marrow progenitor as we understand with hematopoietic stem cells that proliferate, differentiate into different progenitors that are also proliferating, and differentiating into all the blood cell lineages that are circulating. Also, within this compartment, the circulating cell compartment, we have feedback mechanisms. We have implemented in the model the feedback mechanisms from the mature cells to their progenitors. So these are the mechanisms that explain why if you train at a high altitude, you will get a higher number of red blood cells in your circulating. So what it means is that with this model we can get the measurements here on toxicity, use the model, and see what's happening in the blood, predict what's going to happen with the blood counts of the patients. That means we can predict cytopenia risk. And prior to clinical trials, this the strategy with a little bit more granularity. What we do is we measure the exposure in the chip, and we measure this very precisely. We don't assume any nominal concentration. And we dose the chip with different concentrations and different schedules. So here you have one drug, the red drug there. Then we stop. We dose another drug, the blue drug. Stop both and dose again the red drug. So, but what we do different strategies to dose the chip. But what we don't do is to try to recapitulate the human PK, because we don't need to do that. That's not needed. We can use differential equations to handle that complexity. And in the chip, what we establish is the relationship between the exposure and the response in the progenitors. And we use that relationship to predict what this exposure, that is the clinical exposure in patients after a given dosing schedule, what this exposure is going to do in the human hematopoietic cell system. And with that, we predict all the blood counts in patients, lymphocytes, neutrophils, reticulocytes, hemoglobin, at the different schedules. So we can select those schedules that maintain efficacy but decrease toxicity by modifying, just alternating the drugs, or introducing breaks or holidays in the drug administration. And the important thing is that we can do this before clinical trials have started. So does it work? No, that's the big question here. So far it seems it does. So this is an example that Rhiannon has already shown with carboplatin. So platelets, neutrophils, our prediction is the pink line, and the observations are the blue dots, and the black lines are the average and the standard error of the observations. And this is an example with our drugs. So these are active drugs in AstraZeneca pipeline. And so we have here neutrophils, here hemoglobin. And you can see our prediction is the red line, and the observations, well, are the dots, but the blue line and the shadow are the summary, or not the standard error and the average of the observations. So it's not perfect. This for hemoglobin, this for neutrophils, this is just an example. It's not perfect, but I think it's quite powerful to be able to have this red line before you start dosing patients. So, and this just to show you how things have been going in AstraZeneca. In 2019, we, within the group, the number of iTox studies for hematotoxicity using the MPS were quite low, and we were mainly using the rats. In 2022, this has changed, and we are using more MPS studies than rat studies. And that's because we have improved accuracy of our predictions when we use the MPS than when we use the rat. And even if there are failures in the MPS, that there are and there will be, one advantage is that we control the system, and we can even understand the failure, because in this case we are very close to understand this failure, why it happened. While with the rats, the failures are common, and it's very difficult to understand why, so it's very difficult to predict when it's going to fail in the future. So, and just a very, very quick overview of what we are doing to assess GI toxicity. So, just an introduction, because Rhiannon didn't explain this one, and so just a very short introduction on the intestinal epithelium, that is the tissue affected by by cancer treatments. So this is a micro image of electronic image of the epithelium. So you have here the crypts that are invaginations in the mucosa, and these are the villi, protrusions towards the lumen. So, and this is the epithelium covering crypts and villi. You have the stem cells at the base of the crypt. They proliferate, differentiate, and that creates forces that pushes cells upwards, and cells shed from the tip of the villi, so that everything keep balance. To study the epithelium, we have organoids. They are very useful. So here you have an organoid growing, and then they grow as a cyst, and then they develop in the mouse, also in the human, depending on the medium. They develop these cysts that are crypt domains. Also, we have microtissues. So we have systems, in vitro, advanced in vitro systems to assess toxicity in the epithelium. So we are just trying the same approach. So we have dose, we can dose, so we have dose with 5-FU, 5-fluorouracil, that is a common oncology treatment for cancer. And we have dosed the organoids with different concentrations. We measure the toxicity in proliferative cells, in intestinal, in epithelial proliferative cells. And then we have a mathematical model of the human small intestinal epithelium. The model includes stem cells, proliferative cells, differentiated cells, villus cells, and also a feedback to control proliferation from the villus that explains one of the behaviors of the crypt and the villus. And so when drugs and metabolites, what they do is inducing cell arrest or apoptosis in proliferative cells and stem cells, and that creates a deficit in the crypt that induces a deficit of cells in the villus, and that break in the intestinal barrier is what increases diarrhea risk. So here we have, because well, the endpoint for intestinal toxicity associated with cancer treatments is clinical diarrhea. So the endpoint for the prediction is clinical, one of the endpoints is incidence of clinical diarrhea. And so the prediction for this treatment of 5-FU is that, well, you will get like a 35% incidence after the first dose, this 5-FU dose every 2 weeks, then it comes back to baseline, then increases again after the second cycle of 5-FU. And you reach like something like 35% incidence of diarrhea with recovery in between cycles. Well, the clinical diarrhea reported for studies with the same dose of, with the same treatment of 5-FU is 22%. And still we think, I mean, again, we think that is quite a promising result. So in summary, we aim at generating early predictions of drug-induced clinical toxicity. In AstraZeneca, there are multiple functions at the moment, just working on these novel safety strategies to combine mathematical modeling with advanced in vitro systems. And so far, we think the performance of the MPS and the organoids, these advanced cell systems, is superior to the in vivo models, traditional, the rodents. So, well, thank you very much for your attention, and thanks to AstraZeneca colleagues and also TransQST Consortium, that it was a consortium in which part of the work in the GI was developed.
Thank you, Carmen. So now we have a few minutes for questions. So.
Very good talk. I'm wondering how well does your mathematical model fit with the human physiological conditions? The prediction rate is 33%.
I'm not sure I understand the question. Can you repeat it, please?
Yes. So how well does the mathematical models fit well with the physiological condition?
With physiological conditions. How are the mathematical models calibrated?
Yeah.
The mathematical model is, well, the structure comes from the knowledge in biology, and the parameters are also coming from how the human bone marrow works. So there's quite a lot of literature that helps, and also experiments in the in vitro experiments that can help to understand how they work. And then as final refinement, because we have clinical data that shows how the bone marrow is injured, and so the cell counts go down and they recover. So we always refine finally the parameters by matching those observations. So it's a long, the answer is probably we do that very, very carefully, and trying to embrace as much knowledge as possible in literature and in data. It's difficult to generate these models, but once they are generated and working, they are beautiful models.
Are you also working on some artificial intelligence algorithms or the models instead of the analytical models?
So can you repeat again the question? If we also?
Are you also working on some artificial intelligence algorithm models besides the analytic? Because your model is, I understand, is an analytic model, right?
Yeah, these models are mechanistic models but we use artificial intelligence for, kind of data science approaches for the variability, the interpatient variability. That's something very difficult and very difficult to handle with a deterministic approach. So in there we are using data science approach to say, well, this is the expectation, but then we are going to have a distribution of patients around the expectation. And for those distributions, we are using kind of, data science approaches, yes.
Okay, thank you.
Thank you.
Very nice presentation. I was wondering if you are using even a single cell phenotypic analysis to develop mathematical model.
Single cell from?
Phenotypic analysis, like morphology, like other single cell data to develop mathematical model.
Single cell data. Yes. We are using for the bone marrow we use the flow cytometry analysis that is, single cell, you are assessing single cells.
Even information that you can get from
these models are population-based. We have, but we have this because these are ODEs, so this is the average of the cell population. But we have, for the GI, I haven't presented here that model, but we have agent-based model that is single cell focus. And in that one, we really use the single cell data because here we use single cell data just to generate the average response of the cell population. But we have a model, an agent-based model in the GI that is very powerful, and there we really use the single cell data with all its granularity because we have a model representing single cells. So yes, it depends on the approach we use, but yes, we have one model that uses models single cells and uses single cell data input. Yeah.
Okay, thank you.
Okay, so thanks you again, Carmen, and maybe we can move to the next talk and the next.